Good morning. In today's video, we will be covering the topic of proteins. So in the chapter biological molecules, we will be first covering proteins. So we need to know that proteins are the most abundant organic compounds found in the human body and in cells. So the distinguishing factor between fats, lipids, and carbohydrates are that apart from just carbon, hydrogen, and oxygen, protein also contain nitrogen and sulfur atoms. Now, how are proteins made? We all know that polymers of amino acids combine to make polypeptide chains, and when these polypeptide chains combine, they help to form proteins. Now, these are some of the functions written of proteins. However, the most important functions of proteins are the formation of antibodies and clotting, mm, and in the making of enzymes. You can also just read all the rest of these functions. Now, as we talked earlier in our videos as well, that although there are about 170 types of amino acids, the most common amino acids are about 20 to 22 in number, which are discussed here. Now we need to know the basic structure of an amino acid. In the basic structure of an amino acid contains a carboxylic acid group and an amino group, as well as a variable side chain. The central carbon is known as the alpha carbon. Now, how is a peptide bond formed? We need to know that during peptide bond formation, there is always a dehydration reaction going on, in which there is a loss of water. So here is a pretty good example how two peptides glycine and alanine combine to form glycylalanine with the removal of water molecule. In this linkage, CO, carbon double bond, oxygen, NH, this linkage is, you have to remember that this is always the linkage for a peptide bond. Now, we will be covering the different structure levels of protein. There are four structure levels of protein: primary, secondary, tertiary, and quaternary. The primary structure determines the number and the sequence of amino acids. So the primary example is of insulin, which contains 51 amino acids. The secondary form of proteins includes alpha helical structures, including DNA, then they are in a beta pleated sheet. You have to basically memorize the types of bonds and their examples, which are repeatedly asked in MCQs. So you need to know that secondary organization of protein molecules contains hydrogen bonds, and its examples are alpha helix or beta pleated sheet. Similarly, tertiary. So basically, the when the organization increases from primary to secondary to tertiary, the more complex the structure becomes, and so the greater the greater the basically is the bonding and the number of bonds. So when it comes to tertiary structure, you can memorize that tertiary stands for 3D. So it assumes a 3D globular shape, and the bonds which are formed are ionic, hydrogen, and disulfide. The example of tertiary organization of protein molecules is pro- the protein present in muscles, which is myoglobin. And finally, is the quaternary organization. Now, you need to know that in quaternary type of organization is formed by the aggregation of hydrophobic interactions. So it includes hydrogen, ionic, and hydrophobic interactions. Hydrophobic interactions is the key point here. And the main example is the protein hemoglobin, which helps to carry oxygen through our RBCs. Now, this is just the detail of what we discussed in the table above, that insulin contains 51 amino acids. You just need to know that its individual composition as well, that the alpha chain contains 21 and the beta chain contains 30 amino acids. All this numerical information is asked in the MDCAT examination. It contains a lot of rote learning. So now we will be covering, in my video, my aim is just to teach you the concepts. So I'll just be teaching you the conceptual information. And what is a point mutation? A point mutation is the replacement of a single amino acid by another amino acid, and these deleted mutations can lead to the change in the entire reading of the DNA. There are several defects, however, for the purpose of our syllabus, we'll be covering sickle cell anemia in detail. Sickle cell anemia is a hereditary disorder in which the RBCs become sickle crescent shaped. You know that normally RBCs are circular by concave shaped. However, they become sickle cell shaped. So as a defect, they cannot contain adequate amount of hemoglobin, and so the cells soon die. Therefore, sickle cell RBCs are not viable for human life. However, you should know that the sickle cell anemia is more prevalent in Africans since it is basically an adaptation by the human body to prevent against malaria. Now, what happens is that there is a point mutation of a single amino acid. You have to know the amino acid which is involved is glutamic acid, which is replaced by valine. Now, this you have to also have to know this information that in an alpha helix has a uniform geometric structure with about 3.6 amino acids in each alpha helix. Now, I told you before as well that the primary example of tertiary structure is myoglobin, and the primary example of quaternary structure is hemoglobin. These examples are repeatedly tested in the examination, and this is just an example of the molecular structure of hemoglobin, which lies in the quaternary structure. As you can see, it has alpha 1, alpha 2, and beta 1 and beta 2 globin chains, and in the central portion is heme. Now, you need to know that heme contains iron in the center. Now, here are the differences between fibrous and globular proteins. As the name suggests, that fibrous proteins play a role in the structure, fibrous proteins basically have a structural role since they are used to make fibers such as cell fibers and myosin and fibrins. However, globular, you need to know that globular proteins are basically involved in making enzymes, so they have a more functional role. Now, obviously, since fibrous, the functions of the two are opposite to each other. So, since fibrous proteins are elastic, globular proteins will be non-elastic. So you need to know that all of the main functional roles of enzymes, which we talked about, like antibodies and clotting and the making of enzymes, all these are the functions of globular proteins, whereas the rest are the functions of fibrous proteins. Now, these are examples of various proteins, like collagen is used to make bone and cartilage, and histone are used to make chromosome. You can just give them a quick overview reading and you will be able to cover it. Now we will be covering the rest portion of lipids and carbohydrates in our next videos. Thank you. This is all for today. I hope you enjoyed the video. Thank you.